Hey everyone, we are going to go through this Venn diagram together. So when you are done, make sure you take a picture of your completed Venn diagram and submit it into Canvas. This is for cohort B. This is one of your virtual day assignments for the weekend. Okay, so a Venn diagram, of course, compares and contrasts two different things, and the things that are common go in the middle. So we gave you a word bank, and based on the word bank, you should be able to then go through and kind of put them in the appropriate spot. So a couple things when we talk about both of these are both something that we refer to as nucleic acids. So DNA stands for deoxyribonucleic acid. RNA is ribonucleic acid. So nucleic acids as a whole are made up of three parts. They're made up of a sugar, they're made up of a phosphate, and they are made up of a base. These three things collectively make up something referred to as a nucleotide. They make up a nucleotide. Okay, so just kind of keep that in the back of your mind. They make up nucleotides. So sugar, phosphate, base make up a nucleotide. Now, when we talk about the types of sugar, there are two types of sugars. For DNA, the sugar is referred to as deoxyribose. That is the sugar found in DNA, and ribose is the sugar found in RNA. They both have the same phosphate. And then when we talk about these bases right here, the base pairs that we're talking about, the bases are made up of, there's four of them, right? A's, T's, C's, and G's. But however, there's a fifth one that is specific to RNA only. So for every A, there's a T is what we said, and there's an A in both DNA and RNA. For every C, there's a G, and every G, there's a C. Those are the three bases that are common in both. So C's, A's, and G's. DNA has a type of sugar that is specific to it. It is the T, it's the thymine, and RNA has a base that's referred to as uracil, and that's unique to that. So when we do DNA to RNA, there's no such thing as a T, we use a U instead. Okay, so that is your base pairs. When we talk about DNA is what we refer to as the master copy. It's the master copy of the genetic information that never leaves the nucleus. So the master copy is in the nucleus only. DNA never leaves the nucleus. It's the master copy in the nucleus only. Whereas RNA is a disposable copy. So when they're done, we can break it down and destroy it. And this is something that leaves the nucleus. Okay? And that disposable copy will leave the nucleus and it will go and assemble amino acids to make proteins on the, I spelled that wrong, on the ribosome. Okay? On the ribosome. When we talk about DNA and RNA, our DNA model that we made in class we started as a single strand, but we only did the coding side. So when we were done, we made the complement. DNA is a double strand, whereas RNA, especially mRNA, is a single strand. Okay, it's a single strand versus a double strand. But both of these things contain genetic information, okay? So both of these contain genetic information that codes for a message, okay? So that is our compare and contrast ideas. On the bottom here, what we are trying to ask you to do is complementary base pairing. So when we do complementary base pairing, that's Chargaff's rule, and what we're going to start with is DNA to DNA. So with Chargaff's rule, for every T, there's an A. For every A, there's a T. For every T, there's an A. So what I'm doing is I'm drawing the complements, or I'm writing the complements of those. For every C, there's a G, then there's a T, and a C, and an A, right? So for every C, there's a G. For every A, there's a T. For every G, there's a C. For every T, there's an A. Okay? Then we go T, G, G. Then we go T, C, A, T, G, A. Okay, so that's how those, and this is not enough space, but I can't do anything about it at this point, but that's the complement. Hopefully you can write a little bit smaller than I did. So that is DNA to DNA. But when we go through this process from DNA to mRNA, this is something we referenced in class already. This is called transcription. It's the same idea, complementary base pairing. However, there's no such thing as a T. So when we do that, for every A, there is a U. That's really the idea. So CGC is GCG. T becomes an A. For every A, there's a U. For every G, there's a C. For every C, there's a G. For every T, for every A, there's a U. G-A-U, G-C-A-U-G-A-G. Okay? Complementary base pairing still is true, but instead of there being a T, there's the letter U when we go from DNA to RNA. Make sure you take a picture of this and submit your Venn diagram in Canvas.